Hello everyone. I've decided to put this PowerPoint, the second part of our lecture we had on Monday, for you on YouTube, just to make sure that you got everything and that we covered all aspects that you guys need to learn in the human systems. So if we review together the structure of the cell, we will see it's a complete system where you have, if you would compare it to a restaurant or a company or a manufacturing place, you will have the recipe, and we agreed that the recipe will be in the DNA, and that is in the nucleus. The nucleus, as you can see in the picture here, has pores, and the little pores, the little holes that you have in the nuclear envelope, will allow the exchange between material within the nucleus and material outside in the cytosol. Next to the nucleus, you will see a network of membranous structures and tunnels. And these are called endoplasmic reticulum, and you will see some of them are roughened because of the presence of ribosomes, and we call that rough endoplasmic reticulum, whereas others are smooth because they don't have the ribosomes stuck on it, and that's the smooth endoplasmic reticulum. We also agreed in the lecture that the rough endoplasmic reticulum and the ribosomes that are stuck on it are really responsible for making proteins. Your ribosomes are the cooks who will be looking at a copy of your recipe and start to ask someone else to fetch the ingredients so they can start to make the meal, or in this case the protein. So it's really the ribosomes that make the protein or the polypeptide chain, and that protein is going to be modified later on in the endoplasmic reticulum, and also here in the Golgi apparatus. The Golgi apparatus, or the Golgi complex, is the place where the vesicles that got secreted from or released from the endoplasmic reticulum will merge here into the Golgi apparatus, and the final touch will be added at the Golgi apparatus to the product that was initially made here in the rough endoplasmic reticulum. Now you have your final protein in a vesicle-like structure, and those vesicles will be exported outside for exocytosis, and we agreed that the exocytosis is an active transport which involves merging that vesicle that got released from the Golgi into the cell membrane of the cell, and then spitting out the content to the extracellular space in a process we called exocytosis. So we have most of the ribosomes, as you can see here on this graph, are stuck to endoplasmic reticulum, but you will also see some of them are free, and here are some examples of those ribosomes. And the free ribosomes around here are the ones that are responsible for making proteins that are going to stay within the cytosol, within the cytoplasm. Other organelles that we can see, we have the mitochondria, and we agreed that the mitochondria are the places where the energy is mostly produced. It's not the only place where it's produced, but at least it's the big component here is produced through the citric acid cycle and the electron transport chain. We have also the centromere, and the centromere are tubular structure that will help in organizing the microtubules, especially in the cell division. It's present in all the human cells, but it's in some eukaryotes is not present, like some fungi. It's not present. But at least since we're dealing with human system, then it's worth to mention that the centriole are critical for organizing the microtubules, which we will learn about later on as part of the cytoskeleton. So the cytoskeleton has, just like we have bones and the fish has like the bones, the The cytoskeleton is the skeleton that defines the shape of the cell and keeps the structure intact. Also, if there is pulling on the cell, like in the muscle cells, then you would like the pulling to be done on the fibers, on these cytoskeleton fibers, because of course the cell membrane is so weak you can't pull on a cell membrane. If you remember from the lecture, I told you the phospholipids, they're not really bonded to each other. They are sort of fluid nature, just like throwing balls on the top of the filled sink or the filled bathtub. So you want to pull on a structure that holds the tension, and that will be, of course, your cytoskeleton. So we have three types of cytoskeleton fibers. The microtubules, these are the thick ones, and they're made by a protein called tubulin. And then we have the intermediate filaments, and we have the microfilaments. Microfilaments, for example, like actin filaments. Am I missing any of the structures here? We have something called lysosome. And the lysosome is also a vesicle that's being formed here, but instead of being moved to the cell surface where it will go outside the cell in a process we called exocytosis, this particular vesicle is going to stay inside the cell. And it has certain protein coating that will allow the cell to understand that this is not to be exported, this is something we want to remain inside. And those vesicles will contain some digestive enzymes, chemicals that will destroy the target, also acid, just like what you have in your stomach. So in case the cell encounters a bacteria it ate in a process we call phagocytosis, or some structure inside the cell got old enough and you would like to destroy that, and for renewing the structures all over, you would merge that structure, whether you ate or within the cell something dying, with the lysosome. And now the lysosome will liquefy the content of the other vesicle. And so the lysosome will be the equivalent, if you have a business or a restaurant, of the cleaning personnel who will take the trash and will clean the dishes and the tables to make sure that it's ready for the next customers and there is no infection going around. And so they're very, very critical. The very final thing, also another vesicular structure, but this time it's closer to the surface. We call that peroxisome. And the peroxisome, it destroys the oxidative radicals. It destroys chemicals. It doesn't destroy proteins. It has the enzymes that will neutralize oxidants, for example. It has the catalase that will break and neutralize hydrogen peroxide, for example. It also is capable of making chemicals that will neutralize and attack foreign particles. So we will learn that when we cover the immune system and the immune response. But these are pretty much what you have as a summary of the cells and the structures of the cell. So let's talk a little bit more about those, and you will see that we covered pretty much everything in the review slide that I just showed you. So the nucleus here will be the function of the nucleus will be holding the genetic material and controlling the cell division and controlling the cell functions, because it's holding all the DNA, it's holding all the recipe that you will need for making all the proteins that are controlling the cell. The structural features of it will have double layer of nuclear membrane, which is highly similar to the cell membrane, although with a little changes of the protein content of it. That's something we call nuclear pores, and we agreed earlier that the nuclear pores are critical for the exchange of material into the nucleus and out of the nucleus. The DNA is arranged into what we call chromatin, and the chromatin are chromosome, and the chromatin will be divided into chromosomes. And the chromosomes we have in human system, we have 46 chromosomes, and if you remember, 44 plus XY or XX, XY if you're a male, XX if you are a female. The nucleolus is the center of the nucleus. That means the little nucleus, and that's the place where the ribosomes are made. You remember the ribosomes, your cooks? That's where they're made, inside the nucleus. And that's of course another reason we want the nuclear pores, because things that are made inside the nucleus, such as the ribosome, would like that to be exported outside the nucleus into the cytoplasm. So here is the nucleus again, and we can appreciate here the structure of the nucleus. Here is an actual electron microscopy picture, or transmission electron microscopy picture, and we have here the nucleus, and the nucleolus is the center here. And we see here in a drawing how the pores look like. And remember that the nucleolus is the place where the ribosomes are made, right? Okay. So the ribosomes then, these are the place where the protein synthesis will take place, or it's the cook that will start to read the code and that you will provide, and the code is generally a copy of your recipe, so it's a copy of your DNA. And the location, either they are cytoplasmic, that means free, or most of them, as we discussed, they are stuck on the endoplasmic reticulum. And that's why the endoplasmic reticulum will be called a rough endoplasmic reticulum in that case. And so here, speaking of the endoplasmic reticulum, we have the rough endoplasmic reticulum, and because of its association with the ribosome, it's really the place where the proteins are made and slightly modified. The smooth endoplasmic reticulum, on the other hand, has no ribosome, so it has nothing to do with the protein synthesis. However, if the protein is already made at that stage, then the smooth endoplasmic reticulum can alter it, can modify it slightly, can garnish it, can put the spices on it. And it's also the place where you synthesize non-protein chemicals such as lipids, such as hormones that are not proteins. It's also the place where you would like to detoxify the chemicals. You take things orally or by contact or inhalation, like chemicals, like drugs that you have taken, medicine that you took, and you would like all these chemicals to be removed, and the detoxification process happens, the detoxification process happens inside the smooth endoplasmic reticulum. Remember, the smooth endoplasmic reticulum is smooth because it does not have the ribosomes, and because it doesn't have the ribosomes, it does not play a role in making the proteins, but it plays a role in other things, such as making steroids, such as synthesizing lipids, and such as detoxification process, or the detoxifying process. Alright then, so here is a picture of the rough endoplasmic reticulum. If you remember the very original, or the initial picture I showed earlier in the presentation, and here is the smooth endoplasmic reticulum, and we know now the difference, both the functional difference and the structural difference between the two. Now the Golgi apparatus is the next step, and imagine this is the very final garnisher in your restaurant. You made a big meal and now you would like to present it nicely, pack it, because you would like to export it in the smallest possible vesicle. You don't want to make outrageous vesicle that's never going to be excreted outside the cell. So you would like it to be packed, remove all the unnecessary items. And those will be functions of the Golgi apparatus. It modifies, it puts the final touch. It also adds a tag to your protein, because not all the proteins are going to the same direction. So it's essentially putting the zip code on the shipping object in order to direct it to a certain location. Certain proteins inside the cells will be moved to different sites. Some go to the top of the cell, some go to the bottom of the cell, some go to the sidewise, and you would like to know which way is this protein going, and that will be the function of the Golgi, to add that kind of a zip code or a code that will allow the movers, we have movers, believe it or not, inside the cells, to take these proteins and move it toward the target. And that's what we mean by the packaging address, and the shipping address will be added here. Golgi is the name of the scientist who discovered it, and that's of course why we have it with a capital G. And here's a picture of the Golgi. And the way they look like, imagine that you have like flat pancakes or pita breads. Pita bread will be better example because it has a cavity inside. And then the very extreme ends on both sides, it will have a swelling, as you can see here, and those swellings we'll call cisternae. So, but anyhow, you can see here a stack of these pancakes or pita bread on top of each other. And we agreed to the function of them, the final modification of the protein and the final packaging place, and also adds modification and adds a shipping address in order for the cell to understand where are these vesicles going afterwards. Are they going to stay as lysosomes, or are they going to be secreted with exocytosis outside the cell? So that brings us to the vesicles. And the vesicles, these are either storage or shipping vesicles. Things are like packages or moving back and forth inside the cell. And some of them are secretory. That means they are going to be moving outside the cells. These are the export vesicles. And endocytic vesicles, these are vesicles that the cell ate from the surrounding media. And now you have those little things lingering around, and you have to deal with them somehow. Of the vesicles also we have the peroxisomes, and the peroxisomes, they contain enzymes that detoxify chemicals and detoxify free radicals, oxygen radicals. Lysosomes, on the other hand, they contain digestive enzymes, and the digestive enzymes, just like what you have in your stomach, will digest protein. It contains also acidic nature. That means it has a lot of free hydrogen ions, and that lowers the pH. So the lysosomes are very similar to your stomach. They will digest and destroy targets, and those targets can be from the outside, but also targets from within the cell, mitochondria that got old or whatnot. Alright, so these are the structures, and here's what you can see what the lysosome function would be. And here you made the lysosome from the Golgi, and now at the same time you phagocytose the bacteria. Let's say that this is a macrophage, and you phagocytosed here a bacteria. And the bacteria will end up in an endosome, as you can see here, or a phagosome. And that phagosome will fuse with a lysosome. And now the enzymes that you had in the lysosome and the pH you had in the lysosome will merge with this vesicle, and therefore will destroy the content of that endosome, and that will leave you with a mush, and we call that the residual body, and that will be wasted outside. So it's really great, and at the same time the lysosomes also can detoxify and can cleanse something that's dying within the cell, so it's not always something from the outside. On the other hand, the peroxisome will contain chemicals or enzymes, chemicals and enzymes that enable it to detoxify cell waste, toxic cell waste, alcohol, free radicals, and then it will also be exocytosed outside the cells after it has been already detoxified. So finally, the mitochondria. The mitochondria will be the power plant. You cannot operate anything inside the cell without having some power. Now, although the mitochondria are really the place where most of the ATP is made, it's not the only place where ATP is made. ATP can be made also in the cytoplasm in a process we call glycolysis. But the mitochondria will make most of your energy, most of your adenosine triphosphate. But in order for it to do that, it utilizes oxygen, and it will produce carbon dioxide. And that's why we call it oxidative respiration. So it's oxidative feature that relies on oxygen. Without it, you don't have any energy being made at the mitochondria. You will continue to make energy by glycolysis, but not at the mitochondria, which strictly requires oxygen. So here's the structure of the mitochondria. You will see shelves in the inside, and those shelves are in the inside membrane. See how they are made. And that's really to increase the surface area and also to increase the potential space between the two membranes. And we call that intermembranous space. Intermembranous space, the space between the membranes. And so it is this space that you will have the hydrogen ions, as you will see later on, being accumulated, and it's at the inner membrane of the mitochondria that you will generate quite a bit of the ATP. So we will talk about that as we proceed. Where do we get the energy? We get the energy from fat, from sugar. You learned in the chemical nature of particles that you have fat, and the fat can be fatty acids or triglycerides or diglycerides or even monoglycerides. And it produces to you a lot of energy. It's actually producing much more energy, calories per gram. It produces almost double what the sugar will produce to you. Now the sugar is either a monosaccharide like glucose or fructose, or polysaccharide. And the polysaccharide we have in our system, we don't have starch, we're not plants, but instead we have glycogen. And that's your carbohydrate storage. In order for you to utilize it, you have to break it first into monosaccharide, a single sugar like glucose, and then you work your way on that glucose and release the energy from it. Finally, we have other structures inside the cell, and that's the cytoskeleton, for example, the microtubules, microfilaments. We agreed that these go and provide you to support structure inside the cell, and also it provides you the highways and the tracks, the railroad tracks inside the cell for shipping. Remember how I told you certain proteins are shipped into different directions? Now, utilizing this highway, utilizing the tracks that are made by the cytoskeleton, then little particles, tiny little people will carry these proteins and walk on the track to the different directions where the protein is heading. Nothing is random inside the cell. Everything is very organized. Everyone knows where he's going. Now, the cilia are present in some cells, some other cells don't have it. Most of the cells in the human don't have it. But we have cells like the one you have in your nose and in your ear tube. And those cilia are meant to brush things away. So here's a great example where you would like to brush the mucus and you would like to brush all the particles away from your system, and that will be the function of the cilia. We also have them in the tube that is leading from the ovary to the uterus. We call that ovarian or fallopian tube. Now, the flagella is the only flagella we know of in the human system is the sperm. The one that's found on the sperm, and that's a long single whip, sort of like Indiana Jones whip, but instead of making a sound, it propels you like a snake. It propels you forward, and that will be the function of the flagella. The flagella is really made by microtubules, and it's moved by energy, utilizing energy to make this whip. The centrioles, if you remember from my introductory slide, the centrioles are tubular structure inside the cells, and they will be responsible for the organization of your microtubules, and that will be very important in cell division because the mitotic spindle will start there. Alright, so here are some pictures of what we saw of this microfilaments and the microtubules. It doesn't show here the intermediate filaments, but there are also intermediate filaments. All right, so the energy and the summation of all the energy that's happening, we will be dividing those inside the cell. You see many of the things that happen inside. Sorry, I was distracted by the phone ringing earlier. The energy that is being produced, it has to come from somewhere, and you usually, if you want to, let's say you drive the car and the car is not going to drive itself. You want to put gas in it. So what you do is you're putting a chemical energy inside your car and then burn it, so that will give you some kinetic energy. At the same thing, you want to perform some work, so you take a sandwich or you're taking pizza, and that will give you some energy that allows you to do the work. Well, the cell does the same thing. If it wants some energy to perform the work, then it will burn some of the glucose, it will burn some of your fat in order for the cell to produce energy that will allow the cell to make the work. On the other hand, when you don't want the energy, when you have excess of nutrients, then the cell will store those nutrients, and in a process that's building up, not destroying. And the building up process, we will call that anabolism, whereas the breaking up process, we call that catabolism. So we'll start with the anabolism. The anabolism will require enzymes to do so, just like when you are building a house, you are requiring workers to build a house. And you will require energy, because you can't build a bigger structure without using any energy whatsoever, right? And it's used in assembling and building components and energy storing as well. You build your muscles by anabolism, you build fat by anabolism, you put more bone structures by anabolism, but the opposite of which is called catabolism. Now the catabolism also requires enzymes, because you can't just destroy it all of a sudden. You want to release the energy little at a time. And if you remember the example I gave you in the class, that you can release all the energy from oil, for example, by burning it up, but it's a lot of energy released at once. You would like to do it in stepwise release of energy, and so allows you gradual access to the energy that is being stored, and you break down nutrients in order for you to recycle cell components and so forth. Alright, so glucose will be the easiest example for us to harvest energy from. Glucose is not the only energy source. You can take energy from glucose, you can take it also from fat or from protein. Many other sources will produce to you energy as well. But glucose is the simplest and the most ready form to take energy from, in the absence of which you can go to fat and proteins. But as long as sugar is available, then you will use sugar to generate ATP. How do you do so? Well, the first part of it is to work in the cytoplasm, and inside the cytoplasm you will make something called glycolysis, and then later on you will do the oxidative respiration. So in the end you will have from the glucose and oxygen, and if you remember from chemistry, it's C6H12O6. That's the formula for glucose or fructose. Then you add that with oxygen, and then you get carbon dioxide and water. Carbon dioxide and water. Now this process, we don't want it to happen suddenly, because we don't want to burn it and zap your burning everything inside your cells. You want to make it gradually. And the gradual way of releasing the energy will be released in the form of ATP. And the ATP is your hybrid batteries that will hold those energy for you until you need them, and it will be released slowly as you require them for the cellular process. Now the stages of making the cellular respiration is initially the glycolysis, and we're going to talk about these guys. Glycolysis, and then there is a small step between the glycolysis and the citric acid cycle, and then we have the electron transport system. So let's start with the glycolysis, which is this step here. So you take the sugar, glyco, if you remember, means sugar, lysis means breaking. If you remember lysosome, it's the breaking vesicle. Lyso is breaking, right? Or liquefying. So glycolysis is liquefying or breaking up glucose, and so, or sugar, right? So we have glycolysis that will give you energy, and it will give you small energy, but it's enough, especially if you don't have oxygen. So in the absence of the mitochondrial component, or the absence of oxygen, you still are making glycolysis and you're still making a little bit of ATP. Okay? Now, if it proceeds forward, then the product of the glycolysis will enter into a preparatory step, and that preparatory step will lead to the citric acid cycle. We're going to talk quickly about it. And finally, you have what we call electron transport chain or electron transport system, and that allows you to make tons of ATP. So let's start with the glycolysis. In glycolysis, you start with a sugar molecule that's 6-carbon molecule. You break it up into 2 through different enzymatic actions, and you end up with 2 ATP, 2 pyruvic acid. The pyruvic acid are 3-carbon molecules, and you get some energy, which is the 2 ATP, but you also get something that's carrying electrons for you, and these will be your NAD. NAD are the electron carriers. And think of it as someone who will hold your energy for a little bit, but then will provide it to another structure, so the energy from the electrons are not going to be disruptive to the cell. That NAD, once it accepts an electron, then it will become NADH. NADH. Okay? And we'll see that in a little bit. So that's really what takes place in the glycolysis. You have the glucose. It's going to break up into, you need to remember these numbers. 2 pyruvates gives you 2 ATP, and at the same time you have 2 NADH molecules. Why NADH? Because the NAD accepted an electron. In addition, it will acquire hydrogen ions, and that will make it NADH. So we get that. Glycolysis happens in the cytoplasm. It does not require oxygen. It starts with a 6-carbon molecule, and the end product you have 2 pyruvic acids, 2 ATP, and 2 NADH. Sounds good? So if you don't have oxygen, you're going to stop there because you can't proceed any further. If you have oxygen, then the pyruvate is going to go to the preparatory step. At the door of the mitochondria, the pyruvate has to lose a carbon. Once it loses a carbon, it becomes acetyl. And that acetyl is going to be joined with a coenzyme A to make acetyl coenzyme A. Good? Great. So the high energy electrons and the hydrogen ions that are removed, they will be making NADH. So now you have the NADH being formed, and that's also a preparatory step. By the way, this NADH can start to go also into the mitochondria. It can get sucked from the cytoplasm into the mitochondria because we're going to need it in the mitochondria in a little bit. Sounds good? Great. So here's what happened in the pyruvate. So we have the pyruvate will give you carbon dioxide, leaving 1 carbon molecule. The NAD will acquire a hydrogen ion, and now you have NADH. That NADH will get sucked into the space inside the matrix of the mitochondria. Also, the NADH that we made in the glycolysis will be moved into the mitochondria as well. At the same time, after you remove the carbon molecule here, you made an acetyl group. Acetyl group has 2 carbon molecules, has 2 carbon atoms. And those 2 carbon atoms, or the acetyl group, will be joining with coenzyme A into making what we call acetyl coenzyme A. Now, the acetyl coenzyme A can enter to the mitochondria, and it will get into something called Krebs cycle or citric acid cycle. Now, in the Krebs or citric acid cycle, the energy that will be extracted from the remaining 2 carbon molecules, which we call acetyl, is not that much. You will get only 2 ATP. However, most of your energy will be converted into NADH and FADH. So now you're having a little bit of ATP, but really most of the energy will be in the NADH and FADH format. Everything else will be burned into carbon dioxide and water. Or just carbon dioxide, sorry, no water yet. Carbon dioxide will just keep being released from the carbon chain that you introduced, which had 2 carbon chain. Do you remember what we called that? We called it acetyl, right? those two carbon atoms are going to change into becoming carbon dioxide and another carbon dioxide. Now, here's the citric acid cycle. You don't need to remember all the stages here, but you need to appreciate the fact that the acetyl coenzyme A will give the acetyl group, and the coenzyme A will recycle itself to take another acetyl, and so on and so on. It keeps shuttling, sort of like a little shuttle that takes people from the airport, bring them in, dump them at the door, then goes back to the airport, bring more people, dump them, go back, more people, and so on and so on. That's what the coenzyme A will do. Now the acetyl group will combine itself with the citric acid, and that's why we call it citric acid cycle. And little by little we are releasing the energy either in the form of ATP, or we are releasing it in the form of NADH and FADH. Now the FADH and the NADH will hold on the energy, and you will see in a little bit what they are able to do. If we count the NADH, we have 1, 2, 3 NADH and 1 FADH2. In the preparatory step we had another NADH, so that makes 4 NADH and 1 FADH2. Multiply that by 2, then you have 8 NADH and 2 FADH2. Why 2? Because we have 2 pyruvic acid from each glucose molecule, so we multiplied the number we had here by 2. So now the sum, we have 8 NADH, 2 FADH2, combine it with the 2 NADH that we got from the glycolysis, so the total number now is 10 NADH and 2 FADH2, right? 10 NADH and 2 FADH2. Now these guys will hold the energy, right? The electron energy, and will donate it, and you will see what happens when they donate it in the electron transport chain in a little bit, which is right here. So let's see here what the NADH and the FADH are capable of doing. Remember, they were holding energy, right? The energy is in the form of electrons. By the way, electrons is the same energy you have in your electric plug. They're electrons. And so once you zap a molecule with these electrons, the molecule will get excited. And here we have the NADH donating its electrons into this protein. And when you give those electrons to the protein will get excited. And when it's excited, it's going to dump the hydrogen. It's going to take hydrogen ion and pump it across this wall against this mitochondrial membrane into the intermembranous space. The FADH2 is the same thing. It's going to lose the electrons, and when it loses the electrons, it's going to lose the hydrogen. Then it becomes FAD, and this guy becomes NAD. Where did the electrons go? They excited this protein or excited that protein. When you excite those proteins, then you are pumping the hydrogen into that hydrogen ions in that space. And the same thing when the electron reaches this guy, you also are pumping more hydrogen. You keep pumping the hydrogen this way, and now you have huge amount of hydrogen ions being built. Very acidic nature indeed in the intermembranous space because of all the hydrogens that were being pumped in there. How they got pumped? By the energy. Where did the energy come from? From the electrons. Where did the electrons come from? From the NADH and the FADH2. Where did the NADH come from? From the glycolysis and from the preparatory step and from the citric acid cycle. Where did the FADH2 come from? From the citric acid cycle. And now that energy is going to be utilized to energize these molecules, and therefore you are pumping the hydrogen ions into the intermembranous space. And now what happens is you have high concentration of hydrogen here and low concentration of hydrogen here. That's great. So if you remember the rule of diffusion, Ions would like to go from the area of high concentration to the area of low concentration. So far we have been dumping and storing the hydrogen ions, but the hydrogen ions are getting really crowded here and they would like to go back. There is no place to go back here, no place to go back here, 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 or there, and bam, they find a place to go back to. And when they go back through this place, they will energize this protein just by their movement inside that channel, inside this hydrogen channel. And when they do that, they will energize this molecule, and this molecule is called ATP synthase. It's the molecule that will make ATP, right? And so it will take ADP with phosphate and utilizes the energy now the hydrogen ion gave it into making ATP. That's how most of your ATP in your system is made, by the ATP synthase. Well, some of you are wondering, okay then, what happened to my electrons? Or they keep bouncing around? No, they don't bounce around anymore. What happens is those electrons finally will get stuck to the oxygen. And when they get stuck to the oxygen, then they reduce the oxygen, and the oxygen now can bind freely with the hydrogen ions, and therefore you make water. So oxygen plus hydrogen ions plus the electrons that came from here can make to you the water. If you remember, the carbon dioxide was released at the preparatory step and at the citric acid cycle. No carbon dioxide was released in the glycolysis, but however in the preparatory step and the citric acid cycle we had the carbon dioxide, but we had no water being released. Water is going to be released here, and it's really the oxygen in the water is coming from the oxygen you're breathing. So oxygen plus hydrogen ions plus the electrons will give you the water, and these are the products of your electron transport system. So very briefly again, the NADH and the FADH2 are holding energy. They're going to donate the energy or the electrons into these proteins. The proteins will act as pumps. Pumps for what? For hydrogen ions. We called it proton pumps or hydrogen ion pumps. You're dumping hydrogen ions into this space, and the hydrogen would like to go back to the cell. It doesn't go back except through the ATP synthase. Once it rushes its way through the ATP synthase, then it will energize it, and that's why you're making ATP. What happens to the electrons? It's going to unite itself with oxygen, and the oxygen is going to unite itself with some of the hydrogen ions that are coming in, and therefore you are making water. Well, what if this fails to happen? What if oxidative part of the respiration fails to happen and you're making only glycolysis? Well, then you're making only pyruvic acid, and you're making only very little energy. And the pyruvic acid can't stay forever. It will get reduced into something called lactic acid, and that's what gives you the burning sensation in your leg sometimes after you worked for a long time. It also gives you the sense of fatigue because it affects your brain and makes you feel tired when you have too much lactic acid. Get that? All right. So we have a lot of enzymes that are contributing to the reactions. In the end, if you go with oxidative respiration, you get about 34 or 36 ATP molecules. In the absence of oxygen, then you're going to be making very limited amount of ATP. You're still going to be making ATP, but limited. And at the same time, you're producing lactic acid because lactic acid is being produced from the pyruvate that you were unable to burn completely. So this is the summary of the process again. Glycolysis will give you 2 pyruvates. Pyruvates will be taken to the preparatory step, will give you 2 acetyl groups. It will be taken by coenzyme A to the citric acid cycle. That will give you only 2 ATP, and then you continue moving forward into the electron transport chain, and in the electron transport chain you're making tons of the ATP, and that is really made by the movement of the hydrogen ions from the intermembranous space back through the inner membrane of the mitochondria, and that will energize something we called ATP synthase, and you're making tons of ATP in this way. All right, so you can utilize other energy sources. Sugar is one of them, but you can also use fat. The fatty acids, you can break them and you can make acetyl group, and if you remember what we do with the acetyl group, we combine it with coenzyme A. Once we have acetyl coenzyme A, then you have the ingredient for your citric acid cycle to keep on going. And the proteins also have good energy, and you break it up and can harvest that energy. Fat in general has twice the energy as the carbohydrate, calorie per gram wise. Proteins will have the same energy as the carbohydrate. Okay? So, if, like I said earlier, you don't have oxygen, then you cannot have cellular respiration. You will only have glycolysis, and the pyruvate will build up. Pyruvate will be converted into lactic acid, which will give you the burning sensation in your leg. And the 2 ATP you will be making will be the only thing you will be making, which is a very little energy, but it's better than nothing. Once oxygen is available, the lactic acid will be metabolized aerobically, and then to give you carbon dioxide and water. Okay, so in the absence of oxygen, this is not going to be allowed, but only this part. The glycolysis will continue, giving you little ATP and pyruvate. We agreed that the pyruvate will be converted into lactic acid. Sounds good? So, this is a summary of the second part of the lecture. We were sort of running out of time, so I wanted you to get the full advantage of having the lecture by posting it for you online. And I will continue to do that with you. I will try to post the lectures ahead of time, so it will allow us to have the lectures for more discussions and hopefully more questions from you. Sounds good? So I hope you will find this video useful, and I look forward to working with you throughout the rest of the semester. Have a good day.